Hello grade 12s, welcome to our channel once again. My name is Veleleni Nkosi. In today's video, we will talk about blood grouping and the dihybrid crosses. So this is part of genetics, life sciences, grade 12. So this document that you see on your screen is an examination guideline. So this is the content of this video. I will show the different blood groups or the blood type, and then I will solve a problem involving inheritance of blood type, and then in dihybrid cross I will explain the Mendes principle of independent assessment, and then I will solve one dihybrid genetic problem, and then in the problem I will show you the proportion and the ratio of genotype and the phenotype that are produced when we solve dihybrid problem. So this is the content of this video. Without wasting more time, then let's get to it. So here are the blood group. So we have four main type of group. We have type A, we have type B, we have type AB, and then we have type O. So what makes different between this blood type is something called a molecule that we called antigens. So type A contains antigen A, while type B contains antigen B. So this is what makes different. And then type AB contains both antigen A and antigen B, and then type O has no antigen. So this is the phenotype of the blood group. So you must know that the what makes this blood different are the antigen. And then next up we talk about alleles. So there are four blood types in humans. So we have type A, type B, type AB, and type O. So these phenotypes are controlled by 3 alleles, but each person still inherits 2 alleles. So that is mean everyone got alleles from mother and then alleles from father to determine the blood type. So here we've got alleles. So alleles are represented by capital letter I and then superscript A, or capital letter I and superscript B, and then lower cap I. So for capital letter I and superscript A, this one is the allele for blood type A, and then this one is for allele for blood type B, and this one is for allele for blood type O, because this one is empty. So and then blood type A and blood type B are both codominant. Blood type O is recessive to both A and B. So this I, if you've got a genotype that has I and then is either having I and then A or B, so what we will express will be A or B. So you must know that this one is recessive. Remember, recessive is masked by the dominance. So A and B are dominants to O. So for example, here is the table that is explaining. So here we have a genotype for AA, then we end up with blood type A, or the phenotype is A, and then if it's A and the small letter I, we still have A. Also with B, if it's B, and then we have blood type B, and then if it's I and then B, we still got blood type B, and then here we've got A and B. So because A and B are codominant, then we have AB because they both expressed. And then if you've got a genotype with I and then I, we will end up with blood type O. This is the blood type O. It's only be expressed if you've got 2 recessive alleles. So if you've got the dominant allele, blood type O, it's not expressed. So the people with blood type O, they only have recessive alleles. So this is how we differentiate the alleles. This is how we determine the blood type. So we use alleles to determine the blood type. So next up, let me do a problem involving inheritance of blood type. So a man and a woman both have a blood group B. Use a genetic cross to show how it is possible for them to have a child with blood group O, because they both have a blood group B, but how is it possible that they can have a child with blood group O? And then from the previous table, we know that blood group B, it can be formed if we use IB and then IB, they will make a type B. And then again, if we have IB and then O, or a allele for O, we'll also have a type B. So these are the only time we will have blood type B. So in this example, we will cross these two. So like here is our crossing table. So P1, the phenotype, we have type B. We will cross type B because they both parents contains type B. We cross B and then type B. And then for genotype, we will take this one, still make will produce the phenotype B, and then I B, then I, this one is also produce the genotype B. Here is B, not pi. So, and then this genotype, they will undergo meiosis, or the process of segregation. We will have I B and then I, then crossing with this. These are the gametes, then I. So these gametes, we will put them on this or Punnett square. Here we will have IB and then I will have IB and then small letter I. So now we fertilize this, or we multiply this. So we'll have IB and then IB, and then we'll have IB and then I. This side we will have I and then I, while this side we have IB and then small letter I. So for genotype, these are the genotype of the kids that they are produced, or the child. So the child, we will have 3 type B and then 1 type O. So that is mean they can have a child with blood group O. So this one, we will have type O. So here we showed that 2 parents with type B, they can have a child with type O. So, and now let's see how to use a blood type for paternity testing. So for usage of blood type, blood group are compared for paternity testing. That is mean the blood of the child and the blood of the possible father are compared. And then if the blood group do not match, then the man is not the father. And then if the blood group match, there is a possibility to be the father. Then other testing are conducted. So blood type are used to eliminate the fathers, but are not determining the father, because you will find that many men have the same blood type. So but if the child and the possible father blood are not matching, is not the biological father. So this is how we use paternity testing using blood type. So this process only used to eliminate the father, but it does not determine the father. If the blood are matching, then further testing has to conduct it. And then now let's look at dihybrid crossing. So dihybrid crossing involved 2 pairs of alleles representing different characteristics. Like here, I have homologous chromosome which have 2 alleles. So we have allele for eye color and then allele for height. So like blue, we have blue color, we have brown color, and then we have tall guy and the short guy. So the genotype is B, and then TT, which is, and then the phenotype is tall and then brown. So if we write the genotype there, so let's write the genotype, have B, have T, and then T. And then here we have the law, the Mendel's law of independence. So the law say alleles of the different gene move or segregate independently of each other into the gamete. So what they mean here is if these chromosomes undergo meiosis, the alleles for eye color will not affect the alleles for height, or the alleles for height will not affect the alleles for eye color. So they are independently segregated. Like here, what will happen, the gametes that we will have here, we will have B and then T for, and then B and then T for recessive, and then here, then this one, and then this one, and this one. So the gametes we will have B tall, and then we'll have another gametes for B and then short, while we have a B for blue and then tall, and then B for short. So this is the number of gametes that we will have. We will have 4 gametes. So but they segregated separately. That is mean there is no way eye color will affect the height, or maybe shade the height or whatever. But now let's make example of dihybrid crossing. So the example here is an example. It's a in a pea plant, the allele for tallness T is dominant and the allele for shortness T is recessive. The allele for purple flower is dominant and the allele for white flower is recessive. 2 plants heterozygous for both tallness and purple flower were crossed. So here is the information. We have a phenotype. The phenotype, they say it's tall, and then purple. So phenotype is tall and then purple, and then another thing, they say it's heterozygous. So they don't have the same allele. So for genotype we have T and then T, because tall is dominating, and then we have recessive, and then we have P for small letter P. This is the genotype, and then this is the phenotype, and then this is the phenotype, T, T, and then PP. So they are crossing 2 plants of the heterozygous of both. So these 2 plants are crossed. So now let's see how it we used. So phenotype, it's tall and then purple and then tall and then purple, tall purple plant, and then genotype. So the genotype we say is T, T, and then P, P. So again this side is T, T, and then P, P, and then they will undergo a meiosis, or they will undergo the process of segregation. So if they undergo the process of segregation, that is mean we will have a T, P, and then T, P. We'll have T, P, and then T, small letter P. Again we will have shortness, and then purple, and then we have shortness, and then white. So again this side we will have a T, P. This is the process of, so here are the gametes, and then I will put the gametes in this box here. I will put T, P, T, P, then TP, and then I'll put these gametes in this row. From then I will fertilize them. So here are the genotype for first filiate generation. Here is the genotype, and then for, so here you just have to memorize, if we cross 2 heterozygous plant or 2 heterozygous whatever animals, what we will have 9 of dominating allele. So that means we will have 9 tall and purple plant. So we'll have 9 tall and purple plant, and then we will have 3 of dominating and the recessive. So we have 3 tall and dominating color, recessive color, it's white. And then again we have 3 purple, short, which is recessive, and then dominating color is purple. And then we will have 1 recessive alleles. So we will have 1 short and then white. So here we just have to memorize. So this is the ratio. We have 9 dominating alleles and we have 3 dominating and recessive, and then we have 3 dominating and recessive, and we have 1 recessive alleles. So the ratio it's 9 is to 3 is to 3 is to 1. This is the ratio if they say find the phenotype ratio. So the phenotype ratio is 3 is to 1. It's 9 is to 3 is to 3 is to 1. So this is the phenotype ratio. So